Food had and has always been an important issue in the world. All countries try their best to fulfill the need for food. Agriculture plays important role in the national economic development to achieve food security. Indonesian Agency for Agricultural Research and Development, or known as IARD, Ministry of Agriculture, was established in 1974. Until today, IARD has strategic role to improve agriculture efficiency and competitiveness, as well as contributing significant development on agricultural technology through its innovations. IARD is currently supported by 4 research centers based on commodities. They are food crops, estate crops, horticulture, and animal husbandry. IARD is also strengthened by 7 centers of specified research, 14 research institutions and stations, 33 assessment institutes, with total human resource of 1,680 researchers. 138 among them are research professors. IARD provides quality assurance of its research results through an accreditation or certification process to be acknowledged nationally and worldwide. IARD has produced thousands of innovative technologies, from improved varieties, agriculture cultivation system, fertilizers and pesticides, to advanced technologies such as fertilizing drone or smart greenhouse. Around 600 technologies are the superior ones and are expected to be widely used by the community. Indonesia has a great potential to increase agriculture production. IARD contributes in determining its strategic food commodities, namely rice, maize, soybean, shallot, garlic, chili, sugar, and beef to be priorities for strategic self-sufficient program through innovative technology. Therefore, the national needs shall be fulfilled. Technology implementation from on-farm to post-harvest guarantees an increase in the quality and quantity of agricultural produce. The challenge to meet food needs is not only for the country, but Indonesia can also support the world. IARD will continue to innovate to provide technologies toward Indonesia as the World Food Basket 2045.
University of Mataram was established on October 2nd, 1962. Located at the heart of idyllic island of Lombok, which renowned for its beauty, natural and cultural diversity, make Mataram University poses a great opportunity to explore knowledge. Becoming internationally competitive research-based university, ignite the passion for Mataram University to continue pioneering intellectuals by creatively applying their knowledge in research and discovery. Nurturing talented youth to become future leaders who eager to pursue the truth based on faith and piety, give Mataram University a firm ground to contribute at its best to the society. In 2018, Mataram University is ready to answer the call with 9 faculties, 58 departments, study programs, postgraduate programs, vocational programs, diploma. Faculty of Economics, Faculty of Animal Husbandry, Faculty of Agriculture, Faculty of Law, Faculty of Teacher Training and Education, Faculty of Engineering, Faculty of Mathematics and Natural Science, Faculty of Medicine, Faculty of Food Technology and Agro-Industry, Study Programs, Communication Studies, International Relations, Sociology. Mataram University mission in optimizing research and discovery, enhanced with facilities and accessibility for the students and researchers, as well as stakeholders with same common goals in supporting education to solve humanity problems, to fulfill our social responsibility. Hand in hand with the society, Mataram University never to choose run alone. We prefer to walk together through collaboration and joint cooperation with partners from every corner of the world in exploring knowledge, science, and technology. Mataram University commit to find the best way in increasing society's quality of life. We have tasted the same hard times. We have swallowed the same sour times. We have passed the storm of challenge and stay strong.
Sebagai Sekda Provinsi Nusa Tenggara Barat,
Ketua DPRD Provinsi Nusa Tenggara Barat,
Direktur Jenderal Prasarana dan Sarana Pertanian, Kementerian Pertanian Republik Indonesia.
saya sangat bangga melihat perkembangan Universitas Mataram yang begitu maju.
saya merasa bangga menjadi bagian dari Universitas Mataram.
Dan kami sebagai Ketua DPRD mendorong agar Universitas Mataram sebagai sebuah universitas berbasis riset sesuai dengan visi misinya pada tahun 2025.
Saya yakin Universitas Mataram akan mampu meraih visi dan misinya.
Universitas Mataram sebagai salah satu perguruan tinggi yang mumpuni untuk mempersiapkan kader-kader penerus.
Assalamualaikum warahmatullahi wabarakatuh.
Science Innovation Network. Kami hadir mengikuti perkembangan penelitian peternakan dunia dan selalu berupaya menghasilkan inovasi untuk negeri. Keindahan bumi Indonesia yang membentang dari Sabang sampai Merauke dengan kekayaan sumber daya genetik hingga pelosok Nusantara. kaya akan sumber daya genetik ternak, beserta sumber bahan pakannya, menjadikan kami senantiasa menghasilkan inovasi peternakan dan veteriner untuk negeri ini. Bersama kami, Pusat Penelitian dan Pengembangan Peternakan, Badan Penelitian dan Pengembangan Pertanian, Kementerian Pertanian. Telah menjadi saksi sejarah dunia penelitian peternakan dan kesehatan hewan di Indonesia selama puluhan tahun lamanya dengan berbagai inovasi yang dihasilkan demi kemajuan peternakan dan kesehatan hewan bangsa Indonesia. Pusat Penelitian dan Pengembangan Peternakan merupakan unit kerja Eselon 2 di Kementerian Pertanian di bawah koordinasi dan bertanggung jawab langsung kepada Badan Penelitian dan Pengembangan Pertanian, didukung oleh 4 unit pelaksana teknis, yakni Balai Besar Penelitian Veteriner, Balai Penelitian Ternak, Loka Penelitian Kambing Potong, dan Loka Penelitian Sapi Potong. Dengan tugas utama adalah melaksanakan penyusunan kebijakan teknis, rencana dan program, penelitian, pengembangan dan inovasi di bidang peternakan dan kesehatan hewan, serta pemantauan, evaluasi, dan pelaporan pelaksanaan kegiatan. Puslitbangnak senantiasa aktif menghasilkan yang terbaik untuk kemajuan peternakan dan kesehatan hewan di Indonesia. Didukung oleh sumber daya manusia yang kuat, cerdas, dan kompeten di bidangnya. Terus menghasilkan inovasi peternakan dan kesehatan hewan, mengutamakan kebutuhan peternak, serta selalu memberikan pelayanan terbaik, menghadirkan beragam kemudahan bagi peternak melalui kemajuan teknologi. Kami berkomitmen menghasilkan inovasi hasil penelitian dan pengembangan di bidang peternakan dan kesehatan hewan sebagai prioritas dengan upaya diseminasi yang merata di seluruh Indonesia. Bekerjasama dengan mitra secara profesional dan sesuai prosedur, berintegritas, serta menguasai, mengembangkan, dan membagikan pengetahuan dengan stakeholder. Menjaga kualitas dan kuantitas hasil penelitian dengan mendaftarkan di hak kekayaan intelektual maupun paten bagi inovasi yang dihasilkan.
Insya Allah penginten ku abdi ieu hayam bade diurus dienaukeun naon nami-nami penghasilan abdi
untuk telur kon telur lah. Nah yang lelakinya untuk dijual.
Ya rasanya ya enak lamun dipahi mah wong dipahi jang usaha menang modal usaha terus pengen mah belajar bari.
Semenjak dikasih kambing murka dari Puslitbangnak 20 ekor, alhamdulillah sekarang sudah menjadi 42 ekor. Dan terima kasih kepada Puslitbangnak
mendapatkan bantuan sejumlah IT 1060 DOD. Dulu masih baru menetas, sekarang sudah produksi mendapat 345 butir per hari.
Semenjak ada bantuan dari dinas mesin untuk penggilingan pelepah sawit, nah itu sangat membantu.
Selama bertahun-tahun lamanya, Puslitbangnak mengkokohkan diri sebagai riset dan pengembangan peternakan dan kesehatan hewan terkemuka dengan penghargaan yang didapat. Kami peduli untuk selalu memberikan informasi dan manfaat bagi peternak sebagai bentuk tanggung jawab kami. Selalu bergerak menatap ke depan, berupaya untuk terus berkarya, mengutamakan kebutuhan peternak dan mewujudkan kemudahan informasi masyarakat. Pusat Penelitian dan Pengembangan Peternakan menuju inovasi tiada henti.
Assalamualaikum warahmatullahi wabarakatuh. Shalom, Om Swastiastu, Namo Buddhaya, peace be upon you. Good morning, ladies and gentlemen, and best wishes for all of us. Ladies and gentlemen, thank you very much to being with us. The 4th International Seminar on Livestock Production and Veterinary Technology, jointly organized by Indonesian Center for Animal Research and Development and Faculty of Animal Science, University of Mataram. I am Gracia Fatresha as Master of Ceremony at this event. It is great honor for me to host this seminar. Ladies and gentlemen, welcome to opening ceremony of 4th International Seminar on Livestock Production and Veterinary Technology, which is held virtually starting from today, 6 September until 7 September 2021. This platform focuses on technology innovation and collaboration in livestock product for sustainable food system. The theme is facing current livestock condition and highly strategic to deliver our innovation to user. Ladies and gentlemen, during the opening ceremony, I would like to remind you to turn on your video and mute your audio. Before we proceed the program today, I would like to express our sincere gratitude to Our Excellency Indonesian Minister of Agriculture, Dr. Syahrul Yassin Limpo, Excellency Director General of Indonesian Agency for Agricultural Research and Development, Dr. Fajri Jufri, Excellency Director General of the Directorate General of Livestock and Animal Health Services, Dr. Nasrullah, Excellency Rector of University of Mataram, Professor Dr. Lalu Husni, the Honorable Director of Indonesian Center for Animal Research and Development, Dr. Agus Susanto, the Honorable Dean of Faculty of Animal Science, University of Mataram, Dr. Maskur, the Honorable All Director of Research Center under Indonesian Agency for Agricultural Research and Development. Also, I would like to express our sincere gratitude to all invited speakers and participants. Before we begin, I would like to introduce the agendas today, which is 1. Opening, 2. Singing Indonesian National Anthem, 3. Praying, 4. Report by Director of Indonesian Center for Animal Research and Development, following by remark from Rector of University of Mataram. 5. Signing Cooperation Agreement Agenda. 6. Welcoming remark from Director General of IERD, then opening seminar by Indonesian Minister of Agriculture. 7. Traditional dance performance. 8. Plenary session 1. 9. Break and after that, parallel session 1. Ladies and gentlemen, we will sing the Indonesian national anthem, Indonesia Raya. I would like to request all of you to stand up.
Indonesia tanah air
You may have a seat now. Thank you. Ladies and gentlemen, to begin our event today, we would like to have a moment for praying the citizen. Can we please Professor Aji Esdrajat to recite it? The time is yours.
Thanks, Gracie. The Honorable Indonesian Minister of Agriculture, Director Indonesian Agency of Agriculture Research and Development, Rector the University of Mataram, invite speakers, presenters, and participants. Assalamualaikum warahmatullahi wabarakatuh. I will lead you for praying in the Muslim way. Let's up your hand and we pray together. A'udzu billahi minasyaitonir rajim. Bismillahirrahmanirrahim. Alhamdulillahirabbil alamin. Wassalatu wassalamu ala asrafil anbiya'i wal mursalin. Hamdan syakirin, hamdan na'imin. Hamdan yu'afi ni'amahu wa yukafi masyidah. Ya Rabbana lakal hamdu kama yanbaghi li jalali wajhikal karim wa azimi sultanik. Rabbana taqabbal innaka antas samiul alim wa tub alaina innaka antat tawwabur rahim. Rabbana fir lana dzunubana wa kafir anna sayiatina wa tawaffana ma'al abrar. Rabbana fir lana wali walidaina warhamhuma kama rabbayani shighira. Rabbana hab lana min azwajina wa dzurriyyatina qurrata ayun waj'alna lil muttaqina imama. Allahumma inni as'aluka ilman nafi'ah wa rizqan wasi'ah wa shifaan min kulli da'in. Allahumma inna nas'aluka salamatan fid din wa afiyatan fi jasad wa syadatan fil ilmi wa barakatan fi rizqi wa taubatan qablal maut wa rahmatan indal maut wa maghfiratan ba'dal maut. Allahumma hawin alaina fi sakaratil maut wa najatar minan arba'at hindal hisab. Rabbana la tuksik qulubana ba'da id hadaitana wa hab lana min ladunka rahmah innaka antal wahhab. Rabbana atina fid dunya hasanah wa fil akhirati hasanah wa qina adzabannar. Subhana rabbika rabbil izzati amma yasifun wa salamun alal mursalin walhamdulillahi rabbil alamin. Thank you very much. Enjoy your seminar and presentation. Wassalamualaikum warahmatullahi wabarakatuh. Back to you, Gracie. Thank you.
Thank you, Professor Aji. May Allah always give us guidance and help. Amen. Ladies and gentlemen, for the next agenda, we are going to listen the report by Director of Indonesian Center for Animal Research and Development, Dr. Agus Susanto. Please welcome Dr. Agus Susanto.
His Excellency Indonesian Minister of Agriculture, also as the keynote speaker, Dr. Haji Sahrul Yassin Limpo, SHMH, Director General of Indonesian Agency for Agriculture Research and Development, Dr. Insinyur Fajri Jupri, Master Science, Director General of Directorate General of Livestock and Animal Health Services, Dr. Nasrullah MSc, Rector of University Mataram, Professor Dr. Lalu Husni SHMU, all invited speakers, participants as experts, researchers, observers on animal husbandry and veterinary, and also practitioners. Assalamualaikum warahmatullahi wabarakatuh. Good morning, ladies and gentlemen. First of all, let us thank to Allah Almighty for merciful and blessing to all of us, so we can gather in good health and be assembled here in the opening ceremony on the 4th International Seminar on Livestock and Veterinary Technology. On behalf of organizing committee, I would like to welcome you all to this seminar, which is held virtually. A deep appreciation is extended to you, especially to the overseas participants who have been willing to participate in this seminar. As the chairman of organizing committee, I am proud to organize this international seminar on livestock production and veterinary technology, with the main theme is technology, innovation, and collaboration in livestock production for sustainable food system. The theme is facing current livestock condition in Indonesia and highly strategic to deliver our innovation to users. His Excellency, distinguished guests, ladies and gentlemen, as the committee, I would like to report that there are more than 500 participants registered in this seminar. There are 57 submitted papers were invited, evaluated, and selected by the committee from 83 papers. The overseas participants are from universities and research institutions from America, Australia, New Zealand, France, and China, whilst the Indonesian participants come from universities in Indonesia, research institutions under the Ministry of Agriculture, Ministry of Education, Culture, and Research Technology, Indonesian Institute of Science, private institution, and association. There will be 7 invited speakers. 1 come from Indonesia and 6 others from overseas. The Indonesian Minister of Agriculture will give a keynote speech related to the theme of this seminar. His Excellency, distinguished guests, ladies and gentlemen, as it is always the case, nothing in the world is perfect. On behalf of the organizing committee, I apologize for any shortcoming in organizing this seminar. Nevertheless, I would like to thank to the steering committee for the direction and to all members on the organizing committee, both from Indonesian Center for Animal Research Development and University of Mataram for their hard work, which has made possible for us to be here today. Finally, please allow me to request His Excellency, the Indonesian Minister of Agriculture, to give keynote speech and officially open this conference. Thank you very much. Wassalamualaikum warahmatullahi wabarakatuh. Peace be upon you. Thank you very much.
Thank you, Dr. Agus, for the report. And now, I would like to invite Honorable Professor Dr. Lalu Husni, as Rector of University of Mataram, to deliver his remark. For Professor Dr. Lalu Husni, the time is yours.
Bismillahirrahmanirrahim. The Honorable Minister of Agriculture, Dr. Sahrul Yassin Limpo, the Director General of the Indonesian Agency of Agriculture Research and Development, Dr. Insinyur Fajri Jupri MSI, the invited speakers from Australia, New Zealand, USA, France, and China, all presenters and seminar participants. Welcome to the Food International Seminar on Livestock Production and Veterinary Technology. It is my pleasure to welcome you all and to thank you for your contribution to this international seminar. This seminar is important activity for our university because the dissemination of research results is one of our mandates. This is great opportunity of all scientists, practitioners, and students to exchange ideas and learn from each other to further develop our livestock industry. This is the first collaborative international seminar between the University of Mataram and the Indonesian Center of Agriculture, Research and Development, Ministry of Agriculture, Republic of Indonesia. I hope we can continue our collaboration in the future. Please enjoy this academic event, and I hope you can benefit from the interactions between scientists from many different countries participating in this seminar. Thank you.
Thank you, Professor Lalu Husni, for the remark. Now, we move on to signing of the cooperation agreement agenda between Indonesian Center for Animal Research and Development and Faculty of Animal Science, University of Mataram. With honor, I would like to invite Dr. Made Sriasi from Faculty of Animal Science, University of Mataram to guide this agenda.
Thank you, Gracie. Distinguished guests, there are still many challenges and obstacles in the livestock sector that we face in an effort to achieve food self-sufficiency and security. These conditions certainly require the existence of technological innovation to solve problems and perhaps other serious challenges in the livestock industry that will arise in the future. In addition to technology innovation, collaboration, and synergy between various parties, such as the government, academia, businessmen, and the community and the media are also very much needed. Therefore, today there will be a signing of the cooperation between Indonesian Center for Animal Research and Development, ICAD, and Faculty of Animal Science, University of Mataram. From the ICAD side, it is represented by Dr. Hewan Agus Susanto, MSc, as the Director of ICAD, and the Faculty of Animal Science is represented by the Dean, Dr. Insinyur Maskur, MSc. Now, I would like to invite the Director and the Dean to sign the cooperative agreement formally. To the Director and the Dean, please proceed for signing. Please give a big round of applause, everyone. Thank you, Dr. Agus and Dr. Maskur. And we all hope that this is a good starting point for collaboration and the planned collaboration will go well. Gracie, I return the time and opportunity back to you. Thank you.
Thank you, Dr. Agus Susanto and Dr. Insinyur Maskur. And a lot of thanks to Dr. Made Sri Asi for guiding signing cooperation agenda. Ladies and gentlemen, it is now my deep pleasure to please Dr. Fajri Jufri, Director General of Indonesian Agency for Agricultural Research and Development, to deliver his welcoming remark. Dr. Fajri Jufri, the time is yours.
His Excellency Dr. Sahria Silipo, Minister of Agriculture, all invited speakers from Australia, USA, France, New Zealand, China, as well as from Indonesia. In particular, I welcome the participant international seminar. Assalamualaikum warahmatullahi wabarakatuh. Good morning, everyone. First of all, let us thank to Allah, Almighty, for merciful and blessing to all of us. So this morning we are able to attend the international seminar of livestock production and veterinary technology. Good morning, ladies and gentlemen. Indonesian Center for Animal Research and Development is one the center within the IRD with the text of production invention and innovation related to livestock and animal health. One their mandate is to generate new breed of livestock and new variety of forests which has superior productivity and will adapt to the changing climate. Not only that, research is within the coordination of ICRD and encouraged to obtain supporting technology for better livestock productivity, such as animal health, nutrition, and reproduction of poultry and ruminant. Ladies and gentlemen participants, once the invention has been produced, it is time to deliver to many stakeholders, such as local government, private sector, industry, as well as smallholder farmers. Those inventions in livestock and animal health are meant to support the food system, especially from the livestock subsector. Those inventions too will be used to welfare the smallholder farmers, because on the backbone, the production of meat, eggs, and milk are relied on. Since 96% of our animal-based protein sourced or produced by smallholder farmers. Distinguished ladies and gentlemen, this international seminar is very challenging opportunity, and if we need to meet great colleagues over the world. Throughout this occasion, we are delighted to have collaboration and cooperation with other institutions to strengthen of output and impact. Let us work together to the strengthen of capacity and ensure that we'll build better toward productive food system for the nation. Ladies and gentlemen, at this very precious occasion, I look forward to hearing your discussion and learning from you insight. Finally, please allow me to request His Excellency, the Indonesian Minister of Agriculture, to give keynote speak and official open this seminar. Assalamualaikum warahmatullahi wabarakatuh.
Thank you, Dr. Fajri, for the welcoming remark. Honorable distinguished ladies and gentlemen, next we have special keynote address delivered by Honorable Dr. Syahrul Yassin Limpo, Indonesian Minister of Agriculture, as well as to officially opening this seminar. Kindly please, Dr. Syahrul Yassin Limpo, the time is yours.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Salam sehat dan salam sejahtera untuk kita semua. Yang sama saya hormati para undangan, para pembicara, dan pemengkala, praktisi, jajaran Litbang, dan para guru besar, dan sivitas akademika perguruan tinggi yang sempat hadir di tempat ini yang sama saya muliakan. Andang wa syukran lillah, wala haula wala quwata illa billah. Rabbi sholli sodri wa yassir li amri wahlulatan min lisani yafqahu qouli jalal yasiran min ahli. Pertama-tama mari kita panjatkan puji dan syukur kehadirat Allah Subhanahu wa taala yang selalu memberi rahmat dan karunia-Nya sehingga pada hari ini kita dapat hadir dalam acara seminar internasional teknologi peternakan dan veteriner, di mana kegiatan seminar ini merupakan agenda tahunan dari Badan Peneliti dan Pengembangan Pertanian atau Balitbantan, dan pada kali ini diselenggarakan bermitra dengan Universitas Mataram. Terima kasih kepada Pak Rektor dan segenap sivitas akademika Universitas Mataram yang telah mau bersama-sama dengan Balitbantan untuk menemukan upaya-upaya bersama menghadirkan kehidupan pertanian yang besok harus lebih maju, lebih mandiri, dan lebih modern. Di tengah kondisi pandemi COVID-19 yang memasuki tahun kedua ini, saya tentu dan kita semua bergembira bahwa kegiatan ilmiah ini masih dapat diselenggarakan secara virtual maupun fisikal untuk menemukan cara-cara yang terbaik keluar dari lilitan COVID yang tentu saja belum kita tahu kapan ini berakhir, seperti apa kedalamannya. Tapi yang jelas, pertanian, peternakan, dan lain-lain dalam kehidupan kebangsaan harus menjadi kekuatan bangsa ini untuk bisa membangun kehidupan yang besok bagi rakyat yang 273 juta orang itu bisa lebih baik. Saudara-saudara dan hadirin yang sama saya hormati, kegiatan seminar internasional pada tahun 2021 ini dengan tema teknologi, innovation, and collaboration in livestock production for sustainable food system, saya anggap penting untuk diselenggarakan dalam rangka menyampaikan hasil-hasil penelitian, serta sebagai sarana tukar-menukar informasi dan pengetahuan terkini secara nasional dan global. Saya yakin kita pastikan bahwa kemajuan sains, riset, dan teknologi di sekitar kita, di kehidupan kita, sudah begitu berkembang. Ini hanya bisa efektif kalau kita bicarakan bersama, akan menjadi challenge untuk kita temukan cara-cara yang tepat bagi kehidupan, khususnya kehidupan bagi pertanian dan peternakan yang ada di Indonesia. Bapak, Ibu, dan hadirin sekalian, tahun 2021 adalah masa penuh makna. Mengingat kondisi perekonomian global dan nasional kurang menguntungkan kan. Antara lain disebabkan pandemi COVID-19 yang diperkirakan masih akan berlangsung sampai dengan tahun 2022, bahkan mungkin lebih. Dan berbagai sektor memberikan kontribusi terhadap pertumbuhan ekonomi, maka sektor pertanian dinyatakan masih memberi pertumbuhan yang tidak pernah negatif, bahkan berkembang cukup baik di atas 4%. Semester 2020 ini telah menunjukkan tren-tren pengembangan pertanian yang cukup kuat menopang kehidupan negara dan bangsa ini. Seiring dengan wabah COVID-19 yang mulai kita rasakan makin terkendali, diharapkan ekonomi Indonesia dapat bertumbuh dan lebih positif dari kuartal 2 2021 ini. Oleh karena itu, pembangunan pertanian dengan penuh upaya harus diarahkan dalam mewujudkan kondisi yang lebih maju dari apa yang telah kita capai. Pertanian maju diartikan dengan meningkatnya kualitas sumber daya manusia pertanian yang mampu meningkatkan produktivitas dan kerja untuk menunjang kesejahteraan para petani. Sedangkan pertanian mandiri, kita bisa artikan bahwa pembangunan pertanian berdasarkan kemampuan dalam negeri yang disesuaikan dengan kondisi masyarakat kita. Pertanian modern diartikan bahwa pembangunan pertanian berbasiskan inovasi yang sejalan dengan revolusi industri 04. Bapak Ibu sekalian, hadirin sekalian yang saya hormati, suksesnya pembangunan pertanian berkelanjutan tidak terlepas dari peran nyata petani dan peternak karena menyumbangkan sekitar 95% produk pangan bagi 273 juta penduduk Indonesia. Pembangunan korporasi petani di wilayah sumber ekonomi pangan diharapkan mampu memberi posisi kuat bagi para petani. Korporasi petani merupakan suatu model kelembagaan kerjasama ekonomi sekelompok petani dengan orientasi agribisnis melalui kondisi lahan menjadi satu hamparan terkorporasi, tapi dengan tetap menjamin kepemilikan lahan masing-masing petani. Diharapkan melalui korporasi petani, pengelolaan sumber daya lebih optimal karena dilakukan secara lebih terintegrasi, konsisten dan berkelanjutan, sehingga terbentuk usaha yang lebih efisien, efektif, dan memiliki standar mutu tinggi mendorong pertumbuhan ekonomi yang tentu saja berkontribusi kuatnya ekonomi mulai dari pedesaan. Sejalan dengan terbangunnya korporasi petani, maka penerapan digitalisasi akan meningkatkan posisi petani, baik dalam penyediaan input produksi, pemasaran hasil, maupun networking dengan pihak pengelola pangan yang ada. Bapak hadirin sekalian, para narasumber, para expert, dan keluarga besar Kementan yang saya hormati. Dari seminar ini saya mengharapkan dapat dihimpun gagasan, pemikiran, teknologi terbaru yang tentu berkontribusi dalam mempercepat pembangunan pertanian berkelanjutan sehingga visi pertanian Indonesia dapat terwujud. Para kolega akademisi dari luar negeri yang bergabung dalam seminar internasional ini diharapkan saling bergandengan tangan untuk membangun iklim riset yang update, riset yang mampu kita terapkan dalam implementasi lapangan yang lebih efektif dan lebih masif. Hadirin, Bapak, Ibu, dan saudara sekalian yang saya hormati, saya sekali lagi menyambut baik dan menaruh harapan besar dari hasil seminar ini. Mari kita bersama-sama berusaha dengan sungguh-sungguh, tidak mudah berputus asa, dengan penuh keikhlasan, membangun pertanian Indonesia dari subsektor peternakan yang diharapkan dapat mewujudkan kedaulatan pangan hewani bagi bangsa yang besar ini. Insya Allah, Allah akan senantiasa memberikan petunjuk, solusi terbaik, memberikan rahmat dan ridhonya untuk kita semua. Amin. Selamat berlakukan seminar, dan dengan mohon perkenan dan ridha Allah, dengan mengucapkan Bismillahirrahmanirrahim, Seminar Internasional Teknologi Peternakan dan Veteriner tahun 2021 ini saya nyatakan secara resmi kita buka bersama. Ridha Allah bersama kita. Sekian. Wassalamualaikum warahmatullahi wabarakatuh. Saya Sarul Yasin Limpo.
Thank you, Mr. Minister, for the opening this seminar. Ladies and gentlemen, as our welcome greeting, I would like to present a traditional welcome to symbolize our dedication to welcome all of you. It is performed in special traditional called Zapin Rantak Bersambut from West Nusa Tenggara. Please welcome Zapin Rantak Bersambut. Thank you for Zapin Rantak Bersambut performance. Ladies and gentlemen, we welcome you to have a 10-minute break. The next agenda is plenary session 1. Please enjoy the short break time.
Science Innovation Network. Kami hadir mengikuti perkembangan penelitian peternakan dunia dan selalu berupaya menghasilkan inovasi untuk negeri. Keindahan bumi Indonesia yang membentang dari Sabang sampai Merauke dengan kekayaan sumber daya genetik hingga pelosok Nusantara. Kaya akan sumber daya genetik ternak beserta sumber bahan pakannya menjadikan kami senantiasa menghasilkan inovasi peternakan dan veteriner untuk negeri ini. Bersama kami, Pusat Penelitian dan Pengembangan Peternakan, Badan Penelitian dan Pengembangan Pertanian, Kementerian Pertanian. Telah menjadi saksi sejarah dunia penelitian peternakan dan kesehatan hewan di Indonesia selama puluhan tahun lamanya dengan berbagai inovasi yang dihasilkan demi kemajuan peternakan dan kesehatan hewan bangsa Indonesia. Pusat Penelitian dan Pengembangan Peternakan merupakan unit kerja Eselon 2 di Kementerian Pertanian di bawah koordinasi dan bertanggung jawab langsung kepada Badan Penelitian dan Pengembangan Pertanian. Didukung oleh 4 unit pelaksana teknis, yakni Balai Besar Penelitian Veteriner, Balai Penelitian Ternak, Loka Penelitian Kambing Potong, dan Loka Penelitian Sapi Potong. Dengan tugas utama adalah melaksanakan penyusunan kebijakan teknis, rencana dan program, penelitian, pengembangan dan inovasi di bidang peternakan dan kesehatan hewan, serta pemantauan, evaluasi, dan pelaporan pelaksanaan kegiatan. Puslitbangnak senantiasa aktif menghasilkan yang terbaik untuk kemajuan peternakan dan kesehatan hewan di Indonesia. Didukung oleh sumber daya manusia yang kuat, cerdas, dan kompeten di bidangnya. Terus menghasilkan inovasi peternakan dan kesehatan hewan, mengutamakan kebutuhan peternak, serta selalu memberikan pelayanan terbaik. Menghadirkan beragam kemudahan bagi peternak melalui kemajuan teknologi. Kami berkomitmen menghasilkan inovasi hasil penelitian dan pengembangan di bidang peternakan dan kesehatan hewan sebagai prioritas dengan upaya diseminasi yang merata di seluruh Indonesia. Bekerja sama dengan mitra secara profesional dan sesuai prosedur, berintegritas, serta menguasai, mengembangkan, dan membagikan pengetahuan dengan stakeholder. Menjaga kualitas dan kuantitas hasil penelitian dengan mendaftarkan di hak kekayaan intelektual maupun paten bagi inovasi yang dihasilkan.
Insya Allah penginten ku abdi ieu hayam bade diurus diendaokeun naon nami-nami penghasilan abdi
untuk telur kon nelor lah. Nah yang lelakinya untuk dijual.
Ya rasanya ya enak lamun dipai mah wong dipai jong usaha menang modal usaha terus pengen emang belajar bari.
Semenjak dikasih kambing burka dari Puslitbangnak 20 ekor, alhamdulillah sekarang sudah menjadi 42 ekor. Dan terima kasih kepada Puslitbangnak
mendapatkan bantuan sejumlah IT 1060 DOD. Dulu masih baru menetas, sekarang sudah produksi mendapat 345 butir per hari.
Semenjak ada bantuan dari dinas mesin untuk penggilingan pelepah sawit, itu sangat membantu.
Selama bertahun-tahun lamanya, Puslitbangnak mengkokohkan diri sebagai riset dan pengembangan peternakan dan kesehatan hewan terkemuka dengan penghargaan yang didapat. Kami peduli untuk selalu memberikan informasi dan manfaat bagi peternak sebagai bentuk tanggung jawab kami. Selalu bergerak menatap ke depan, berupaya untuk terus berkarya, mengutamakan kebutuhan peternak dan mewujudkan kemudahan informasi masyarakat. Pusat Penelitian dan Pengembangan Peternakan menuju inovasi tiada henti.
Persoalannya memang Prof ada dua, pertumbuhannya yang sangat memang terbatas tapi punya daya tahan. Yang kedua memang dagingnya keras. Kita buktikan malam ini Pak Gub.
Maka pada malam hari ini kami mencoba mempersembahkan ke hadapan Bapak Ibu sekalian spesial Bali daging asli sapi Indonesia yang diharapkan dapat mendunia dan dapat menjadi kebanggaan bangsa Indonesia. Dengan berbagai metodologi yang kita terapkan dalam beberapa tahun terakhir ini, insya Allah Pak Menteri bisa membuktikan nanti dagingnya menjadi lebih empuk.
Untuk pertama kali mendapatkan sapi Bali dengan daging yang selembut ini, saya rasa sudah seperti sapi-sapi yang ada dari impor atau dari luar. Saya tertarik dengan spesial Bali beef ini karena saya menangkap ada aroma khas dan ini mungkin yang kelebihan dengan yang lain. Oleh karena itu saya yakin daging ini, daging Indonesia, punya khas yang Indonesia. Saya yakin ini akan menjadi sebuah prospek bagi kita semua ke depan.
Justru yang kita jual itu keunikannya, nilai nutrisinya, tapi yang kita perlu promosikan itu adalah sapi Bali sebagai daging yang rendah lemak.
Nah, jadi kami menguji banyak sekali formula pakan dan kami menemukan bahwa ada dua tanaman lokal yang ada di daerah kita. Yang pertama adalah daun turi yang ada di banyak di Lombok Selatan dan yang kedua adalah lamtoro.
Perbedaan daging sapi Bali yang kita treatment dengan lamtoro ini, satu adalah kampukan. Dan kedua, memang ada rasa yang spesifik yang tidak ada di daging-daging lain. Ada taste yang berbeda di dalamnya. Saya bisa merasakan perbedaannya.
Akan ada pemain besar yang akan mengembangkan sapi ini sehingga bukan mustahil dalam waktu tidak lama lagi sapi beef Bali yang rasanya tidak kalah dengan sapi-sapi dari Jepang akan menjadi tuan rumah di daerahnya sendiri.
Kita akan buktikan bahwa daging kita ini kalau kita budidayakan akan mampu memberi nilai tambah yang tinggi, itu satu. Yang kedua, dagingnya juga secara khusus bisa dikatakan tidak kalah dengan daging lain. Ya, saya kira alasan untuk impor untuk kita juga sangat tidak masuk akal gitu. Believe your dream and your dream become true. Believe it, make it happen.
Menteri Pertanian Syahrul Yassin Limpo menyerahkan 20.000 DOL chicken atau DOC ayam Kampung Unggul Balitbangtan kepada Menteri Koordinator Perekonomian Erlangga Hartarto. Penyerahan dilakukan secara simbolis saat keduanya menghadiri acara ekspos hasil inovasi Badan Penelitian dan Pengembangan Pertanian di Balai Penelitian Ternak Ciawi Bogor, Kamis 6 Mei 2021. Di kesempatan ini, Erlangga juga memberikan nama untuk galur ayam Kampung Unggul Balitbangtan atau KUB ini.
Jadi saya kasih nama Janaka. Soalnya ini ada Kunti, Ibu Kunti. Janaka itu nama lain dari Arjuna. Satu telur tiap 2 hari. Dan ini bisa juga menjadi pedaging. Dan dengan demikian ini tentunya baik untuk dikembangkan, punya nilai ekonomi yang tinggi.
Menteri Syahrul menilai kehadiran Janaka merupakan salah satu bukti bahwa akselerasi ekonomi di bidang pertanian cukup tinggi. Untuk itu pihaknya siap dalam mengembangkan serta menyebarluaskannya ke berbagai daerah di Indonesia.
Pak Menko tadi memerintahkan pada kami di jajaran Kementerian Pertanian agar ini bisa dikembangkan lebih kuat, bisa disebar ke seluruh pelosok. Saya kira di Idul Fitri ini, Ramadan ini, tanda-tanda akselerasi ekonomi di bidang pertanian cukup tinggi, sejalan juga dengan hasil triwulan 1 PDB nasional kita, kontribusi pertanian naik 2,9.
Janaka sendiri merupakan galur ayam KUB 2 yang telah memiliki perbaikan performa dari ayam KUB 1 melalui seleksi genetik. Performa yang ditingkatkan antara lain produksi telur, sifat mengeram, dan warna sheng atau kaki.
Janaka ini kelebihannya adalah dia mempunyai produksi telur 200 butir per ekor induk per tahun, atau rataan hendainya itu 60%. Tujuan dari pembentukan galur ini adalah sebagai galur unggul female lain, ya, galur betina, untuk menghasilkan DOC ayam kampung unggul, ayam lokal potong. Jadi bisa panen pada umur, kalau yang KUB 1 dulu umur 70 hari, bisa berkurang menjadi sekitar 55 atau 60 hari. Jadi keunggulannya itu. Kemudian sifat mengeram dari indukannya masih ada sekitar 5%.
Selain ayam, pada kesempatan ini Menko Erlangga juga memberi nama untuk sapi Belgian Blue Cross. Sapi yang kemudian diberi nama Pari Kesit ini merupakan hasil persilangan antara Belgian Blue dengan sapi Friesian Holstein yang memiliki rata-rata berat potong sebesar 533 kg. Hingga saat ini, sapi Belgian Blue Cross yang telah lahir sebanyak 22 ekor. Salah satunya berumur 3 bulan 2 minggu yang memiliki bobot ternak 110 kg. Dari Ciawi Bogor, Andika Bakti, Indra Fauzi mengabarkan.
Ayam Kampung Unggul Balitbangtan atau KUB merupakan ayam lokal unggul tipe petelur hasil inovasi teknologi Badan Litbang Pertanian, dalam hal ini Balai Penelitian Ternak. Saat ini populasinya telah berkembang dengan pesat serta menyebar ke seluruh Indonesia. Penyebaran ayam KUB saat ini semakin meningkat setelah dilaksanakannya program bekerja, di mana ayam KUB didiseminasikan kepada masyarakat secara masif. Dalam budidaya ayam KUB, pakan menjadi sangat penting, di mana biaya pakan menjadi biaya produksi terbesar dari seluruh biaya produksi yang dibutuhkan. Melihat akan hal tersebut, Balitnak Balitbangtan memandang sangat diperlukan suatu program layanan untuk para peternak ayam KUB yang dapat membantu para peternak agar dapat membuat pakan ayam KUB secara mandiri. Dengan harapan, peternak dapat menyusun formulasi pakan ayam KUB berbasis bahan pakan yang banyak tersedia di wilayah mereka dengan harga pakan relatif murah. Seiring masuknya era digital 4.0 saat ini, di mana pelayanan publik institusi pemerintah sudah mulai banyak menggunakan teknologi digital untuk mempermudah proses pelayanan terhadap publik. Berdasarkan latar belakang tersebut, Balitnak Balitbangtan membuat suatu inovasi teknologi guna memberikan pelayanan pembuatan formulasi pakan ayam KUB dengan memanfaatkan teknologi digital berbasis Android yang disebut Smart Feed Agrinak. Aplikasi Smart Feed Agrinak atau SFA versi 1.0 dibuat oleh Balitnak Balitbangtan untuk menyusun formulasi pakan ayam KUB pada berbagai fase umur produksi. Aplikasi ini diharapkan dapat membantu peternak agar mampu memformulasikan pakan KUB sendiri berbasis bahan pakan yang tersedia di wilayah sekitar mereka secara mudah hanya dengan sentuhan jari saja. Untuk membuat formulasi, tekan menu formulasi pakan, kemudian pilih jenis umur ayam yang diinginkan. Pilih jenis bahan sumber energi konvensional ataupun inkonvensional dengan cara menekan kotak sebelah kanan pada setiap bahan. Setelah memilih bahan pakan sumber energi, lalu tekan selanjutnya untuk memilih bahan pakan sumber protein dengan langkah yang sama. Dengan langkah yang sama, pilih bahan pakan sumber mineral dan suplemen pakan. Masukkan harga per kilogram setiap bahan yang dipilih, lalu tekan hitung. Sejak dikenalkan pada bulan Agustus 2019, sudah ada lebih dari 2.000 orang yang mengunduh aplikasi ini. Semoga aplikasi ini lebih menginspirasi dan memotivasi para pelaku usaha peternakan ayam lokal untuk menyusun ransum secara mandiri. Diharapkan biaya pakan menjadi relatif lebih rendah, efisiensi tercapai, dan peternakan ayam lokal lebih maju. Jayalah pertanian Indonesia.
Ayam KUB atau ayam kampung unggul hasil inovasi Badan Litbang Pertanian Kementerian Pertanian ternyata telah banyak diadopsi oleh para pelaku usaha ayam taliwang di Lombok, Nusa Tenggara Barat. Ayam KUB merupakan prospek yang cukup menjanjikan dan mampu meningkatkan ekonomi dan pendapatan masyarakat. Meski terkendala pandemi COVID-19 ini dan berdampak pada penurunan permintaan ayam KUB di sektor bisnis rumah makan, Namun, permintaan ayam KUB masih tetap ada untuk digunakan sebagai sajian khas ayam taliwang di Lombok, Nusa Tenggara Barat. Yuk kita simak liputannya.
Kalau menjadi peternak ayam KUB sebenarnya tidak terlalu lama sebenarnya Pak. Tapi berangkat dulu dari hobi, saya mulai dari beternak puyuh dulunya Pak. Peternak puyuh itu ternyata agak berat sebenarnya. Kami mulai melirik sebenarnya seperti apa, tertarik dengan ayam KUB ini seperti apa. Setelah saya lihat perkembangan ayam KUB itu cukup bagus. Selain dia unggul, pertumbuhannya lebih cepat daripada ayam kampung, juga kalau menjadi usaha rumahan atau untuk membantu ekonomi rumah tangga itu luar biasa sekali menurut saya, karena interval dari mulai kita beternak yang kadang-kadang pada saat permintaan pasar itu tinggi, kita bisa 1 bulan saja kita sudah bisa menjualnya, sehingga kita tidak khawatirlah dengan pasarnya. Nah, itu.
Alhamdulillah pada saat ini sampai sebelum pandemi juga emang permintaan untuk ayam KUB ini sangat tinggi, dan itu menjadi kendala kita terkait masalah permintaan yang begitu besar dari konsumen. Kami sebagai peternak, ya disinilah kita untuk memanfaatkan situasi itu, yang memberikan kepada teman-teman peternak yang ada di Kabupaten Lombok Tengah ini, mari di sini kita punya potensi yang besar untuk melakukan usaha ini untuk memperdayakan masyarakat terkait masalah hasil dari presentasi yang akan kita dapatkan terkait masalah permintaan. Keunggulan dari ayam KUB, kenapa peternak NTB ini mengunggulkan atau mengandalkan bahwa ayam KUB ini sebagai pensuplai dari bahan baku usaha kuliner ayam taliwang ini. Yang pertama yaitu dari interval waktu yang cepat. Yang kedua nanti bisa yang jelaskan terkait masalah itu. Dan yang kedua, ayam KUB ini bukan hanya bisa mensuplai dari dari permintaan ayam taliwang, tapi bisa ayam KUB ini juga bisa mensuplai dari warung Padang kayak gitu. Ya, artinya dari ayam KUB ini bukan hanya fokus di taliwang saja, cuma dia bisa kita suplai 2, warung Padang sama warung taliwang kayak gitu.
Beberapa tahun terakhir ini kita sudah merilis beberapa galur seperti contoh ayam KW, ayam kampung Banalim sejak dirilis tahun 2012 ini sudah menyebar di seluruh Indonesia. Banyak sekali provinsi, mulai dari Sabang sampai Merauke tentunya. Jadi di Sumatera, di Kalimantan, di Sulawesi, di NTB ini sudah sangat berkembang sekali. Salah satu contohnya ini ayam taliwang yang terkenal di NTB itu, ini sebagian besar sudah menggunakan bahan dasarnya itu dari ayam KW kita. Ayam KW ini punya keunggulan ini. Dia punya potensi telur 160 sampai 180 butir per tahun. Ini ayam kampung. Ini hasil seleksi dari berbagai jenis ayam kampung yang ada di Indonesia. Jadi umurnya itu pada saat umur 20 minggu itu sudah menghasilkan bobot bahan itu 800 sampai 1 kilo. Jadi dia mulai dipanen itu, dan harga break-even point-nya ini kalau kita melihat hari ini kurang lebih modalnya itu kurang lebih 27 sampai 30.000. Jadi masih sangat, kalau kita jual tentunya ayam kampung kan per kilo di atas 50.000 ya. Dan ini sangat menguntungkan untuk masyarakat peternak kita mengembangkan di seluruh Indonesia. Dan kita mengupayakan memang pakannya ini berdasarkan ketersediaan pakan yang ada di setiap wilayah provinsi. Jadi kalau wilayah itu sentra sawit, ya kita pakai inti sawit, atau sentra kedelai kita pakai kedelai atau pakai jagung. Kita mulai pakai menggunakan pakan in situ yang ada di setiap provinsi atau daerah seperti itu.
Halo Sobat Ternak, hari ini kita mau ngepoin teknologi terbaru dari lolit sapi, diagnosis kebuntingan dini. Pasti penasaran kan? Yuk ikutin saya. Assalamualaikum Bapak.
Waalaikumsalam, Mbak.
Lagi apa ya Pak?
Ini lagi lihat-lihat, vitaminnya kurang. Tapi nggak apa-apa, kenapa tidak?
Bapak nggak usah bingung. Sekarang ada teknologi baru, semua yang dari lokal penelitian sapi potong. Tes kit kebuntingan. Ini Bapak.
Oh ya.
Beberapa keunggulan tes kit ini adalah tidak menimbulkan traumatik, mudah diaplikasikan, dapat mengevaluasi kegagalan kebuntingan lebih awal.
Kami terbantu mengetahui informasi keberhasilan perkawinan lebih awal pada ternak sapi, sehingga dapat menekan biaya produksi dari kegiatan deteksi kebuntingan dini yang dilaksanakan di kota provinsi.
Diharapkan sapi dapat beranak 1 tahun sekali dan populasi sapi potong di Indonesia dapat meningkat, mendukung Indonesia menuju lumbung pangan dunia. Dadah, sampai ketemu lagi di teknologi lolit sapi yang berikutnya.
Excellencies, ladies and gentlemen, now we will begin the first plenary session, who will be led by our moderator, Professor Research Ismet Inonu from Indonesian Center for Animal Research and Development. He is a senior researcher on expertise of animal geneticists, and he also active as a chief editor Indonesian Journal of Animal and Veterinary Sciences. Without further ado, please, Professor Ismet, the time is yours.
Okay, thank you, Grace. Good morning, ladies and gentlemen. I hope that we have a good connection for all session for this morning, because come up something from the internet connection. I am honored to be the moderator for this session with three distinguished invited speakers. The speakers present here are first Professor Dr. Heather Burrow from the University of New England Armidale, and second Dr. Bieng Riel Min from USDA Agriculture Research Service Conservation and Research Laboratory, Texas, USA, and the third is Dr. David O. Callahan from University Montpellier, France. For the first speaker, you will listen to the presentation from Professor Dr. Heather Burrow. She will present her paper entitled Opportunities and Challenges to Increasing Productivity in Cattle Farmed by Smallholders in Asia and Africa. Before that, please allow me to read her CVs. Heather Burrow is a professional research fellow from the University of New England. At present, she is the project leader of Indo-Beef projects operating in Indonesia, funded by Asia. Her work field are quantitative geneticists in beef and dairy cattle. She strongly advocates multi-organizational and multidisciplinary and multinational collaboration. I want to remind all the participants that you write your question at the chat room before the session, and I will read the question at the end of the session. Please welcome Professor Burrow. You will have 20 minutes for the presentation, and we will have the question and answer at the end of the session. Please.
Thank you very much for this presentation, and I am just going to load my screen and hope that you can all see that. So thank you very much for the introduction, and thank you also for inviting me to participate in this seminar. The title, as you have indicated, moderator, is Opportunities and Challenges to Increasing Productivity of Smallholder Cattle Farmers. And the title actually says in Asia and South Africa, in Asia and Africa rather, and there are very specific reasons why I have chosen to look at those two continents. And so just to put this into context, we are looking at the world population. In 2011, there was around 7 billion people, but by 2050, that population is expected to increase to 9 to 10 billion. But most of particular importance, most of that growth is expected to be in Africa. Here we can see an exponential growth in Africa and in Asia, where we are seeing growth continuing perhaps to 2040 and then stabilizing. But it means that the population growth is greatest in Africa and Asia. So if we're going to feed the world by 2050, there is a need to double the outputs from our existing resources, or in some cases, fewer resources, in spite of several major global challenges. You already know of climate change, increased urbanization. They are described in the paper, so I'm not going to go into it here. But because of pressures on agriculture in the advanced economies, most of that increased production actually needs to occur in the regions of the greatest population growth. In other words, in Asia and Africa. So the opportunities for agricultural improvement are greatest for extensive livestock production systems and those in tropical areas. So that is good for both Africa and Asia. Those opportunities use land resources that have few other alternative uses. These types of areas and systems are also able to capitalize on the strengths of ruminants like beef and dairy cattle and sheep and goats, because they all digest lower quality pastures that are not suitable for humans or for monogastric livestock species like pigs and poultry. So if we are looking primarily at cattle, we need to look for cost-effective and transformational technologies. Our cattle need to be very well adapted to their production environments. And in Asia and Africa, these represent cattle that are primarily owned by the smallholder farmers. So traditional technologies aimed at delivering production increases in cattle will assist. So I'm talking here of traditional animal health, traditional animal nutrition. They're going to help, but they're not going to double the outputs that will be needed to achieve food security. Other technologies, non-animal production type technologies will help, such as improved meat and milk processing, improved product shelf life, but I'm not even going to consider those here. I'm focusing only on those that are related to animal production. So what technologies will be available for impact by 2050? And I'm highlighting here impact. The reason being that it usually takes at least 5 to 10 years from initial usage of a technology to achieve practice change and impact on the ground. So what we're looking for here are technologies that are going to be ready for implementation perhaps by 2040, because any later than that, they're not going to achieve the doubling of productivity that is needed by 2050. So these are my personal picks, and you know, they very much are in my area of interest, and no doubt there are others that I'm ignoring, but my picks are genomics and the related omic technologies. There's lots of those technologies. Information and communication technologies, including the spatial information and robotic technologies. And I've added in a third one of artificial reproductive technologies, not because I think that these technologies are going to achieve widespread impact, but because there is potential for some components of that to really achieve strong productivity improvements in those smallholder farmer herds. I've also added in a fourth one because to encourage the practice change, not only with these three new technologies, but also the traditional technologies, we need to find some way of encouraging the farmers to use those proven technologies. And so I'm going to touch on that very, very briefly at the end of my talk. So what do I mean by genomics? You're probably all familiar with the DNA double helix. And so we're using DNA information or genomic information from plants, from livestock, from parasites and the pathogens that affect livestock, and rumen microorganisms. And I'll talk you through these in the next few slides. So what do we actually mean by genomics? Well, to start with, I'm talking about a single nucleotide polymorphism, which is just one example of a DNA technology. And what we have in this slide is in each of these columns is a different animal. And this is actually a figure of some DNA markers from animals, from cattle. And I'm asking you to look particularly at the two top rows here, because what we have are examples of markers that are inherited from the sire or the dam. So every animal has two copies of a marker, one inherited from the sire and one from the dam. And in this example, we see two animals that have two copies each of the favorable form of a DNA marker for meat tenderness, which is called calprotectin. In this example, we have animals that have two copies of the tough form of the calprotectin gene. And in this example, we have we have one copy of each of the favorable and the unfavorable gene. So they have different copies of the gene. And this technology was what began the genomics revolution, if you like, where it was believed that we would be able to identify associations between one of these SNPs and a single trait, and with the aim of genetically improving the trait. As the research has developed, though, we've realised that this form of an association between one SNP and a single trait does not happen. This is known as marker-assisted selection, and generally marker-assisted selection no longer works, it never worked, but we now realise that it doesn't work to the extent that we thought it would initially. So what we've been able to do is to use multiple SNPs, and these are what we call SNP chips. They're examples of different SNP chips. This SNP chip has about 7,000 SNPs on it, 50,000 SNPs, and close to 800,000 SNPs. And we use those smaller SNP chips to impute up, using bioinformatics, to the 3 billion base pairs, which are what we're looking at here, the genomic sequence, T, G, C, and A. And so what we're looking for is to get as close to the full sequence as we can. And the applications of this technology are for livestock breeding and selection, for customized livestock management, and also for new treatments. I will run through these two here in a bit more detail, but I will just mention briefly the new treatments. So vaccines, in this example here, we have a heat image of a tick biting into an animal hide, into a cattle hide, and the genomic sequence of the tick and the cattle were used to be able to identify antigens to develop anti-tick vaccines. The other uses are to increase production and reduce costs and reduce methane emissions. So there's a whole host of treatments that could be developed. I think the most topical at the moment is that genomic information that was used to develop the anti-COVID-19 vaccines. So very shortly after discovery of the COVID-19 virus, the virus was fully sequenced and made available to research groups around the world. And so shortly afterwards, we ended up with vaccines that are now helping to curb the transmission of the COVID-19 pandemic. So in terms of livestock genetic improvement, we have SNP-based tests, and these are the single traits. So for example, the polled and horn gene, but most of the productive and reproductive and adaptive traits are under the control of hundreds or thousands of genes. And for that purpose, we use genomic selection. And we use genomic selection to develop or use existing prediction equations and test them in their local livestock breeds and the reference populations to improve the traits under the control of those multiple genes. And this can be used for both straight bred and crossbred animals. And so in Indonesia, I'm hoping that in the short term, through international collaborations, Indonesia will be able to develop reference populations that are larger populations that can then be linked to international populations in similar production regions in the tropical areas to capture the benefits for Indonesia, but also provide benefits for all the collaborating partners. What we need to recognise is that what works for animals also works for plants. So here we have an example of psyllid-resistant leukemia or Lamtoro, and that type of technology, so the genomic information can be used to develop new plant species. And it can also be used for management applications, particularly to customize animal management. For example, we can sort animals for the different markets or the most profitable markets, or we can manage animals differently if they are known to be either resistant or susceptible to parasites. And these low density or low cost chips could also provide incentives for value-based markets that reward animals with desirable attributes. I've mentioned the information and communication technologies, and they would be used to reduce the cost of production, provide information that's not easily captured by humans, to improve animal management, improve compliance with market specifications, and particularly where data collection applies to individual animals, to improve the rates of genetic gain. So again, just a couple of examples. In Australia, we are using these drones or unmanned aerial vehicles to monitor and measure watering points or ground cover. In Indonesia, we could use drones to monitor canopy cover in palm oil plantations to assess the quality and quantity of pasture that can be grown underneath those canopies. The electronic animal identification, this is just an ear tag or a neck tag that is worn by the animals, and it records an automated individual identity for that animal, and it also records other types of information that I'll give you a couple of examples of. And this is, they are used in conjunction with sensor networks, solar-powered sensor networks that transmit the data to the cloud, and it can then be retrieved using apps on the mobile phones or on iPads. So one example here is a neck collar, and these neck collars are measuring movement of heifers to determine whether or not they are in estrus, and so they can be inseminated at the right time. Another example from Central Australia is a a remote weighing and drafting system. And here the animals are trained to walk through this gateway. They are all individually identified using their electronic tags. We have the solar panel control here, and we have the sensor network there. The animals are weighed, and depending on the weight of the animals, they can be released into this paddock or into that paddock. So this allows automated weaning, for example, if you wanted to wean calves. If the calves are heavy enough to survive on their own, then they're weaned in one way, or they may be underweight animals and they require supplementary feeding. So that's an example of an automated weighing and drafting system. Further development of these types of information and communication technologies are likely to increase exponentially over coming years, and they are going to impact significantly to improve livestock production systems in all countries, not just in Africa and in Asia. So they have huge potential. I've mentioned briefly the artificial reproductive technologies. These are things like cloning, like gene editing, in vitro fertilization. I don't really believe that those types of technologies will have impact by 2015. But it may be possible to modify the current AI or artificial insemination technologies so that improvements to fixed time insemination or development of semen storage at ambient temperatures that allow easier use of AI may have big impact in developing countries. And if the reference populations can be established in Indonesia to achieve genomic selection, those populations could also be used to provide a source of genetically superior semen that will enable the smallholder herds to improve productivity per se. So what I'm saying here is that I'm not expecting the smallholder herds to be doing the genetic improvement. I would be assuming that the reference populations will take care of that, but will provide the semen for use in the smallholder herds. I mentioned briefly the need for commercial incentives. So all of these technologies have strong potential to improve the productivity, but there is no point at all in producing more or higher quality beef for farmers to sell at wet markets, not getting realised value for the product that they deliver. So I see there's a need for new value chains that reward the farmers on the basis of the quality of the product they deliver to market. And to give you one example of this, over the past couple of decades, the Meat Standard Australia beef value chain has provided critical commercial incentives to achieve practice change in Australia. And around 50% of beef carcasses are now graded through MSA with financial rewards to all sectors, to the producers, the processors, the retailers, and the consumers. And so I was very pleased to see the promotional video of special barley beef, because the idea and the thinking behind that special barley beef concept is exactly what we're talking about here. And I think if the special barley beef value chain can be established and shown to be working, that will provide the incentive for a meat standards Indonesia scheme across producers all throughout Indonesia to deliver high quality beef for consumers. So terima kasih, and thank you very much for your attention.
Okay, thank you, Professor Burrow, for your fascinating presentation. Okay, now I want to go to Dr. Dieng Min, the second speaker. Ladies and gentlemen, for the second speaker, you will listen to the presentation from Dr. Min. He will present his paper entitled, excuse me. Start video. Okay. He will present his paper entitled Dietary Mitigation of Enteric Methane Emission and Animal Production from Ruminants, Plant Tannins Mitigation Option. For the moment, please let me read his CVs. Okay. Dr. Bien Real Min got his PhD from the Massey University, New Zealand. At present, he is an associate professor at Tuskegee University. He is an editorial board member in 3 international journals with 9 grants and funded projects, and published 91 articles in international journal. Please welcome Dr. Min. Your time is 20 minutes for the presentation, and we will have question and answer at the end of the session.
Thank you very much, ladies and gentlemen. It is honor to present our research papers today based on our research. Thank you so much for that. I would like to talk about dietary mitigation of entering methane emissions and animal production from ruminants, particularly focusing on plant tannin mitigation option today. Agriculture's Dr. Heather mentioned that our population, global population, will be reached to about 9 to 10 billion by 2050. So to meet food demand for this increasing population, it was estimated that milk and meat production must increase by 63 to 76% respectively. Therefore, livestock industry will play a central role in global economy in the next decade.
Could you make the slide larger?
Sorry?
The slide
Can you hear me?
larger, please. This slide, please make it larger a little bit.
You cannot see?
We can see, but it is small, very small. If you can make it larger, it's better.
Now it's better?
A little bit more.
Hang on.
Okay, okay.
This is,
it's okay. Yeah,
yeah,
okay.
this is a big, sorry. Anyway, however, increasing food production will likely result in an increasing in greenhouse gas emission, including entering methane production from animals, manure, crop production, and fertilizations, unless mitigation practices are discovered and implemented. So I have only 20 minutes, so I'm going to brief introduction to that. Beef cattle and dairy cattle are major global greenhouse gas emission contributors, and buffalo and sheep and goat and other livestock as a minor contributor. How is entering methane produced? Methanogenesis in the rumen during feed digestion, especially including bacteria, protozoa, fungi, and methanogens, they produce methane gas during the digestion. And also this entering methane emission in ruminants, which is produced by fermentation of feed by methanogens archaea, represent a loss of 2 to 12% of gross energy of feed diet. So which is contribute to the global greenhouse gas emission. So most of gas production from forage diet, especially this is a winter wheat forage diet in cattle, they make fructivolate as well as produce of a bunch of gas that including carbon dioxide about 45%, methane gas about 30%, and rest of them is volatile fatty acid gas production. And also it is generally known that ruminants they produce methane gas differently between diet, 2 different diets. Forage-based diet examples produce high methane gas productions, And however, grain-based diet that lowering methane gas emission depending on microbiome population in the rumen. So how can you measure this briefly methane emissions, entering methane emissions? There are three different measurements in this world: respiration chambers, green feed systems, and SF6 systems. And this is in vitro ANCOM gas production system as well. So this is a well-known gas production measurement system at the moment. What kind of factors affecting entering methane emissions? Especially dry matter intake is a major contributor for methane emission. This is average meta-analysis measuring direct chambers, SF6, and green feed system, all of them in dairy and beef cattle. Each 1 kilogram increase in dry matter intake increases methane production by average about 20.5 grams per kilogram of dry matter in beef and dairy cattle industry. So dry matter intake is a major contributor for methane gas emission, also losing energy diet. What kind of thing actually in the dairy cattle, milk production also slightly important associated with the methane gas production. However, most of average daily gain in beef cattle production pronounces more methane gas emission, especially forage-based diet. So increasing dry matter, increasing average daily gain, actually increasing methane gas productions. Also, other rumen fermentation performance also associated with methane gas production, especially acetic acid is highly affected with methane gas inhibition. However, negatively impact for the propionic acid and butyric acid associated with methane gas productions. I don't have much time to do. So today, let's focus on mitigation option for tannin dietary supplementations. What's the story of that manipulation, rumen fermentation using condensed tannin? The condensed tannin containing forages such as crabgrass, mimosa trees. In Indonesia country, they may have a lot of mimosa trees and acacia and cocoa. But in temperate zone, we have perennial ryegrass and Sericales ptejas that contain high level of condensed tannins. Also some fruits and berry like apples, grape, strawberry, blackberries contain some little amount of condensed tannin. Also pine bark also high level of condensed tannin. Also some peanut skin and peanuts contain high level of condensed tannin too. Today, so I would like talking about some of them. So how can we mitigation of methane gas and how can we produce sustainable livestock productions? Firstly, let's see that mode of action of condensed tannins. Once that condensed tannin actually complex form at the neutral pH in the rumen about 6 to 7 and during the chewing and rumination, and then dissociate with the condensed tannin protein complex at acidic condition in the abomination. So using the condensed tannins, we can able to make a bypass protein. So it is possible to make bypass protein from the lumen and potentially be able to reduce methane gas and ammonia production in the rumens. This is one of SDS page gels in evidence that reduce inhibition of proteolysis of white clover fraction 1 proteins. These first figures, there are large and small subunit of protein fractions without condensed tannins. Most of protein levels digested within 2 hour or 4 hour incubations disappear. However, addition of 1% of condensed tannin here, extracted tannin, actually most of large subunit was protected for the proteolysis. But some of them small subunit disappear. So this figure show that addition of condensed tannin in the rumen able to protect and able to make increasing bypass proteins. Here is a rumen gas production. addition of tannin in 0, 1, and 1.5% and 2% in yellow. So increasing tannin concentration in the rumen actually decline of gas production, total gas production in the rumen with a different time basis. And also we added the different chemical composition examples, proxylin, tannin, and monensin. Monensin is one of the popular feed additive for the beef cattle. So increasing time actually increasing gas production. However, addition of tannin and mimosa monensin addition decline of total gas productions. About methane gas production is a similar manner, and then we measure different also Lotus cornicularis forages, which is a temperate zone, tannin containing forages actually declares methane gas and ammonia gas production with depending on how much we added in vitro conditions. How about methanogen bacteria? We using as a model for pine bark diet for the 15 and 30% pine bark diet in meat goat. Actually, methanogen bacteria population is declined. Also, methane gas production is significantly reduced, about 33% methanogenic bacteria reduced, indicating that tannin-rich diet can reduce methane emission in ruminants. Also, most of microbial population, this is a film levels, some microbial films increasing or declines, especially decline of bacteroides and other bacteria. However, especially film of Formicotis population was significantly increasing, indicated that tannin-rich diet potentially manipulate over rumen bacterial community. These figures show that acetic acid and propionic acid ratio versus Formicotis and bacterial film ratio. And the bottom line is average daily gain in meat goat, actually increasing Formicotis population in the rumen, significantly increasing average daily gain. However, acetic acid to propionic ratio decline, actually increasing average daily gain. So rumen fermentation, control of rumen fermentation and microorganism is highly associated with the average daily gain in meat goat. How about methane gas production? This is the peanut skin. We did 3 different lipid experiments. Peanut skin contain high level of tannin, actually increasing tannin concentration in the diet, increasing methanogen population in the rumen. These figures also show that Sericea lespedeza, which means high level of tannin in the forages compared to crabgrass. We measure 12 animal each treatment using open circulate respiration chamber. Actually, addition of feed supplementation of Sericea lespedeza significantly decline of methane gas and ammonia gas as well. So, which is pretty consistent evidence for the methane gas inhibition in ruminants. How about beef cattle? Also, average daily supplementation of condensed tannin, such as no supplementation, 1% and 2% for Baracho condensed tannin supplementation, increasing average daily gain in beef cattle. This is average cumulative body weight changes. Actually, 2% of tannin level in cannulate steers, increasing actually average daily gain as well as live weight changes. This is another experiment for the 70 days mimosa condensed tannin and chestnut condensed tannin extract supplementation, dramatically increasing average daily gain in grazing condition with the beef cattle. This is actually we apply for the pine bark supplementation in meat goat. Pine bark is very popular in United States, which is unlimited natural resources. Also, pine bark is waste, also 11% contain 11% of condensed tannin, also readily sourced. So we grind it or pine bark and make pellet, 0%, 15, and 30% pine bark diet contained in pelletage. And we supplement that control and 15 and 30% pine bark diet. Average daily gain is significantly linearly increasing. Gramera intake is numerically increasing, but not significant. However, gain feed ratio also increasing significantly. So tannin containing diet also very important for the animal productivity, and also decline ammonia concentration in the rumen as well. And carcass productions, we measure hot carcass and cold carcass measurements, actually increasing pine bark concentrations in the diet, increasing carcass yield as well. So, which is excellent figure to find it out. I'm not talking about too much for that fatty acid composition. However, profile for the saturated and monosaturate, unsaturated fatty acid and polyunsaturated fatty acid, and ratio between saturated and unsaturated fatty acid, increasing tannin containing diet, actually increasing mono and polyunsaturated fatty acid in meat goat. In dairy cattle, we measure that in the plasma, tannin containing lotus corniculatus with the 3.6% tannin and 3.9% tannin, actually increasing polyunsaturated fatty acid in plasma, and also omega-3 fatty acid in plasma also increasing. Let's back to the peanut skin again. In indoor experiment with the 22 kiko crossbreed intake male goat, we added control 25 and 50% peanut skin supplementation for 50 days. Actually, they consume more at the 25% peanut skin included, which means that most of meat goat, they love to eat tannin containing diet. Also, average daily gain was most of highest at 25% peanut skin diet, as well as feed efficiency. However, the second experiment with another experiment we conducted that still showed that increasing peanut skin diet and increasing average daily gain. However, this time 50% was little better than 25% of peanut skin included. And so the experiment, we measure with a different peanut skin and without peanut skin. However, low protein versus high protein diet, what happened for average daily gain? Both of them, low and high protein diet with the peanut skin, actually increasing average daily gains. So time is over. So
okay, thank you,
we can
Dr.
say
Min.
that. Sorry.
Yeah, thank you, Dr. Min. And
yeah,
you will see the question in the chat room. Maybe you can read. I'm glad that Professor Burrow is already answered the question. Maybe this will be help some for the time. Okay. Once again, thank you, Dr. Min, for your exciting presentation.
Thank you so much.
Ladies and gentlemen, now you will listen to the presentation from Dr. David O'Callaghan. Maybe you stop the share, Dr. Min. Dr. Callahan, the speaker for this morning.
Good morning.
His presentation entitled Recent Advances in Our Understanding of the Pathogenesis of Brucellosis. Okay, first let me read his CVs. Dr. O'Callaghan got his PhD in 1989 from the University of London. At present, he is the director of INSERM U1047, bacterial virulence and chronic infection, and the French National Reference Center for Human Brucellosis at the University de Montpellier Medical School in Nimes, France. Please welcome Dr. O'Callaghan. You have 20 minutes for the presentation, and we will have questions at the end of the session.
Okay.
Please.
So thank you very much. Firstly, I have to work out how to share my screen. I'll put some glasses on and try and find this. Presentation part.
You want us to...
Where do I share my screen? I can't see anything to share screen. Perhaps I need to chat. How do I share screen? Oh, it's there. Yeah.
Okay.
Yeah, I've modified the slides a little bit since the ones I sent you, so it'd be better to.
Okay. Okay.
But where do I share the screen on here?
It's already there. I see.
It's already there.
Okay. Yeah.
My slides.
Once again. Yeah. Your slide.
Ah, here, a share screen. It's come. Okay, good.
Okay. Okay.
Screen 2. Is that sharing?
Yeah, it is.
No.
Yeah.
No, I can see.
Yeah. It's okay. It's okay. I can see. And
you can see my slides.
Yeah. Put it in the slideshow, please.
Yep. Okay.
Okay.
Slideshow. Okay, you have slideshow because I don't see this on my screen at all now. Do you have the slideshow?
Yeah.
You have the slideshow. Okay, good. And can you see my mouse on the screen? Can you see my mouse pointing, my pointer?
Yes.
Okay.
We can clear. It's clear. Yeah.
Great. Okay, so thank you very much for the invitation to talk to you. As some of you know, I'd hoped to be able to come to Indonesia in person this autumn, but unfortunately it wasn't possible with the pandemic. So hopefully next year we can reorganize this. So I work. So it's a very different type of story here from what we've had in the last two talks. We're going to talk about infectious diseases, and so I work in the medical school here on the top floor at the University Hospital in Nimes, in the south of France. So Nimes is here, right in the very south of France. This is the UK, Spain, and Italy, and we have lots of Roman ruins. So this isn't our laboratory. This is kind of a Roman amphitheater we have in our city. So brucellosis, in fact, it's one of the most important zoonotic infections in the world. So zoonosis is an infection which infects animals but can be passed on to humans. So it's best known as an infection of livestock, and it's passed on to man by either contact with infected animals or consumption of infected animal products, especially dairy products. So in animals, it causes a huge economic problem throughout the world. It causes infectious abortion, which then leads to sterility, has huge problems in livestock trade and livestock movement, and obviously to eradicate the disease in the final stages, infected animals are killed, so there's a huge cost there. And I said it's transferred to humans very, very regularly, and it causes a disease which is known as Malta fever, which is a flu-like disease, but which can become chronic and can of cause very, very long-term infections. It doesn't often kill, but it's a very debilitating disease. So people who've done any microbiology, especially medical microbiology, know about Brucella, and they probably know about these few species here, the first few on the list, which are what we would call the original strains, which have been known since, some of them from about 150, 200 years. Over the last 30 years, the number of species has suddenly rapidly increased, first of all with strains here, starting from cetaceans, from sea mammals and seals and foxes, and then over the last kind of 10 years or so, we've had a huge number of new kind of atypical Brucella which have been isolated. So I'm going to talk to you a little bit now about two things. One about Brucella virulence, how we think it can cause disease in animals and in man, and then talk a little bit about these atypical strains and the challenges they present for kind of identifying these in human and in perhaps animal infections. So work with kind of genome sequence analysis has shown, in fact, that speciations in the kind of classical Brucella is very, very recent. And you can see here, this is what everything has suddenly appeared. These are very, very, very closely related organisms. And then we have this group here of the older, earlier diverging atypical Brucella, which are here. And when we actually look at, so we take these classical Brucella and we look at them in a kind of a more finer detail, we can see we have 7 distinct branches. Each of them, strangely, has a specific animal host. And this actually fits really well with the kind of the classical old-fashioned taxonomy. So genome sequencing has shown that the things that were done by the kind of biochemical assays and things like that in the last 100 years or so are actually very, very good and actually are real. And what the genome sequence analysis has really showed us and confirmed is that Brucella, the genus, is in fact composed of clonal images which result from their co-evolution with their host. This is because the bacteria are actually physically isolated from other bacteria. They don't have an environmental life stage, so there's no chance of any horizontal gene transfer, acquisition of foreign genes from other bacteria, because they're very, very isolated. We just get vertical transmission, which is either intrauterine infection or infection of the young by lactation, and of course, obviously the infection of humans and things like that through eating infected milk, and also consumption of contaminated abortions is also another kind of very clear reason for this transmission from one bacteria from one animal to another. And in some cases, we also get sexual transmission. So you can see there's no real stage where these bacteria are living and growing in the environment. They're always within their animal host, apart from very short times. And some bacteria, some Brucella strains, but not all, are very infectious to non-preferential hosts, such as man. However, in most cases, this is an epidemiological dead end. So a human who has got infected with Brucella is not contagious. He won't pass on brucellosis to another animal, to another person, or to an animal. So we're interested in looking at these genome sequencing to try and work out how did Brucella evolve to become a pathogen. Because actually, it's clear they started kind of many millions and millions of years ago. They started as soil organisms. And the closest related ones at the moment are called the Ochobactrum, for example. And then they went through a period of genome reduction and then acquisition of virulence factors. How they acquire these virulence factors, we're not really sure, because as I said, they don't have an external kind of lifestyle, but they've obviously either acquired virulence factors or developed genes or proteins that they have into virulence factors, which has allowed them to adapt to the kind of the intracellular or intrahost environment. And then they started by causing, the earlier bacteria probably caused very highly acute infections, which would kill the host very, very rapidly. Then we went through a period, and this is kind of many of the atypical bacteria are still like this. They call very acute infections, killing animals very, very rapidly. But then they went through a co-evolution and adaptation with their preferential host and kind of became stealth pathogens. They're not seen, they're just there, kind of just sitting there, living really, almost moving on to into almost a symbiosis type thing. And then we get loss of virulence. Actually, the bacteria are becoming less virulent because they're not causing these acute infections anymore and causing these chronic infections. So As I said, Brucella is a facultative intracellular pathogen. It can live and survive inside host cells. So you can see here, this is with the green. In green are bacteria. The green are bacteria living inside a HeLa cell. After 48 hours, one bacteria has gone in and multiplied to pack the cell solid with these bacteria. In red, you have the late endosomes, which are part of the cell defense system to kill them. And you see this is just kept miles away from it. And also you see there's nothing happening in the nucleus. The nucleus is very, very happy. The EMs is actually a macrophage. So a macrophage is a cell which is there normally for to defend the host against bacteria. And you can see it's packed solid with these bacteria after 48 hours. The bacteria have grown and multiplied and packed the cell solid. So to do this, bacteria have to kind of adapt to life within a host. So we get their kind of first contact with host cells. And in this first contact, they get an idea and invade the cells. Then they have to adapt, establish their life within the host, avoid host defenses. And they do that by adapting to the environment, changing their gene expression to adapt and express the factors that are required to survive in this environment. And then they have to modulate the host biology as well to stop the host killing them and to make the host a place where they want to actually live and where they can live and survive. Then they grow, multiply, can they extend their niche and eventually get out and either move in the environment, which can be perhaps within the blood of the animal, or it can be actually outside of the animal and kind of restart the cycle. And the major virulence factor of Brucella is in fact this thing which we call the VAB type 4 secretion system, T4SS. So a type 4 secretion system is a multi-protein complex which is found in the envelope, in the inner and outer membranes of the bacteria. And this actually works as a molecular syringe, and it's used by the bacteria to inject factors from the cytoplasm of the bacteria into out of the cell. This can be in different ways. It can be originally used for bacterial conjugation, so transfer of DNA from one bacteria to another, or more recently in bacterial killing, bacteria will transfer one protein to another and actually kill other bacteria. Agrobacterium also transfers DNA. Other bacteria, such as Helicobacter, sit on the surface of the bacterium and inject a toxin, the Kag protein, into cells. Others, Bordetella pertussis, uses it to inject or to release its pertussis toxin onto the cells. And then a large number of bacteria, including Brucella, we have Bartonella, we have Legionella, live inside vacuoles within the host cell. And here they inject their proteins out of the vacuole that they're found into the host cell, where they have an effect on the host cell biology. So you can see with Brucella enters through lipid rafts, enters the cell through lipid rafts, and then type 4, it's right very, very early on, the bacteria starts actually secreting these what we call effector proteins, a set of proteins which actually affect the biology of the host cell. They start by affecting vesicular transport. They stop the bacteria being killed by lysosomes and endosomes. And then they allow the bacteria to create the niche in which it's going to grow and live. And they do this by capturing the vesicle with the bacteria in, actually captures vesicles which are being transported from the Golgi to the endoplasmic reticulum. And the bacteria live and grow in a compartment which resembles the endoplasmic reticulum. Then this matures further and we get what we call an autophagosome. We get autophagy which is induced. And this allows the bacteria then to escape and reinfect a new cell. So for 20 minutes, we can't go into a lot of details of this, but just here's a list of some of the Brucella effectors which have been discovered over the last 8 or 9 years really, and really starting to be discovered at the moment and understood now. We're starting to get ideas of what they're actually doing. And we can see we have several classes of effect. We have a large number of proteins which actually affects the biology of the Golgi, of the ER, inducing the unfolded protein response, for example. This actually kind of induces, activates inflammation. It blocks the secretion from the ER to stop things being kind of thrown out of the ER, to kind of extend the place where they want to live. It targets different pathways, and kind of then it slows down the formation of the apoptotic. So this apoptotic BCV, to allow them to stay in this part here as long as possible, keep the cell alive. Others are infecting kind of immune responses in the host. Others are involved in the early stages of the trafficking, and many the effects of them are not known, and we're still trying to work out and understand what they're actually doing. So now let's start talking about the atypical brucella. You can see we have this large number of new atypical brucella which have been discovered, and the real question is, should we worry about the new and atypical brucella? Because the genus is obviously far more diverse than was thought even 20 years ago. And now there are kind of several new species have been discovered every year. So the question is, how many more new brucella species are actually there in the environment? And I think there are kind of a very large number, and especially in kind of exotic environments, jungles, for example, or perhaps even in mangroves, we will find new species affecting different animal species. But the question is, do these strains represent a danger for man or for domestic animals? Because that is the real thing. If they're there and they're not affecting us, then we don't have to worry. So the atypical brucella, they can cause human infections. The first one, there's only been kind of 3 cases so far, so it's still very, very rare. But I think it's perhaps also very underdiagnosed. So this first strain, BO1, Brucella inopinata BO1, was discovered in an abscess or a breast implant. So this is something which is very different to the classical presentation of brucellosis. It's more like kind of one of the later stage symptoms in chronic brucellosis, this abscess. Then BO2 was found in a chronic disruptive pneumonia, again, abscesses. We recently studied a strain called BO3, which again came from kind of multiple abscesses, pulmonary condensations. And you can see here on a scan of the person, you can see the actual infectious foyers within the lungs of the patient. What was very interesting is this strain BO3 was actually very, very similar, almost identical to a strain that we had characterized the year before, which came from a frog, from a Pac-Man frog. And this strain actually grows very rapidly in human cells. You can see here, it grows much faster here. You see the slope here than than a normal brucella. And then what happens is it grows very rapidly and kills the cells. You can see here the cells are packed solid with brucella, which are here in red. So what happens here is, this is again, as I was saying earlier, that in the stages of evolution of brucella, the early brucella were probably far more virulent, and then the brucella that we know have slowly lost their virulence. They're not so virulent because they want to keep their host alive. So this is an example, I think, which shows that these early brucella were perhaps more virulent. The major problem is the question is, are these atypical brucella correctly identified? Firstly, they have very, very unusual phenotypes. Normally, brucella grows very slowly. You get colonies on an agar plate after 3 days, 4 days. Here, these strains, the atypical strains, grow very fast. You get very large mucoid colonies. They have a completely different metabolism. They can use far more carbon sources. Again, this is because they're still undergoing the genome reduction that we had for the in the classical Brucella. And their surface properties, their antigenic properties are different, and they can't be tested by simple slide agglutination tests, which are used to identify the bacteria. And this means that many atypical Brucella isolates are probably misidentified as soil bacteria, such as Ochrobacterium, which are just opportunistic pathogens in humans and just cause kind of much mild infections. They're not normally life-threatening. So this, I said, is a diagnostic challenge. So the problem is that all Brucella were thought to have an O antigen, so on their LPS, which is kind of the surface sugar on the bacteria, which is composed of a polymer pyrosamine. And you can see here, this is encoded by a set of genes which are found in all of the enzymes for this are encoded in a set of genes found in all the Brucella. But in the many of the atypical Brucella, so in the strain BO2, in a frog strain, and also in the BO3 strain that we've recently studied, this region here has been replaced, or is replaced by a set of genes which actually encode an O antigen with a different sugar, rhamnose. And you can see here also on a silver stained gel, we have completely different profiles of LPS. You can see they're completely different. And this means that human infections cannot actually be detected using the standard serological assays that are used in laboratories throughout the world.
Excuse me, please, your time keep.
Okay, I'll be very fast. Other thing, they are also motile. These strains are motile. They can move. They have a flagella. And normal Brucella don't actually have flagella because they have mutations in their genes. And to finish, we'll talk about laboratory-acquired infection. And this is one of the major causes of Brucella is one of the major causes of laboratory-acquired infections in humans. And this is through many things, incorrect manipulation, and also because the machines to identify them are very bad and not working very well. So at the moment, MALDI-TOF mass spectroscopy is revolutionizing bacteriological identification. It means now that you can identify a bacterium in a diagnostic laboratory within about 15 minutes rather than 3 days. Unfortunately, until very recently, the two major sources of manufacturers, the two major machines didn't identify Brucella because it wasn't in their database. So we've worked with bioMérieux and we've actually improved their database, and now bioMérieux the machine will be able to detect Brucella quite clearly and correctly. And another important thing is in terms of safety. For mass spectroscopy, it's very, very easy. You just take a colony from a plate, add it to a little tiny slide, then you add a little 1 microliter of a chemical which will inactivate the bacteria and binds to it. Then you stick it through the mass spec machine. Excuse me. Unfortunately, we found that this process doesn't kill Brucella on the slide. So there's a potential infection risk. So we've modified this procedure, and here we're able to get a new system where we just get the bacteria, actually put them into the solvent first, then add them onto the plate and on the slide, and then test them. And this removes all risk of infection. So this is very important to save for the health and safety of laboratory workers. And so another important thing is that this method actually allows samples to be stored for several days at 4 degrees, and this will allow the bacteria. It means that what can happen is that many mass spec machines are very, very expensive, so this can be done in small laboratories, and they can be safely sent to laboratories with the machines and be identified there. So I think I'll finish there. So thank you for your attention. And this is a list of some of the funding sources.
Okay, thank you, Dr. Karahan. Your presentation is very fascinating, but the time is limited for us. Okay, well, ladies and gentlemen, now come to the time for question and answer for the next 30 minutes. I'm happy that Professor Burrow is already answered the question. But let me read the question, if some left for the session. The secretary will send me the question. No, it's not here. Please, it's already. But I will give the time for Professor Burrow to some more explanation, maybe for 5 minutes. Please, Professor Burrow.
Okay. Yes, I'm. So now I can't see the questions. Just hold on. I'll bring the questions up. So the question that seems to have been generating the most interest is really how we can establish reference populations, because reference populations are relatively large herds of cattle that are measured for all the important traits, so production, reproduction, and ideally adaptation, although the measuring tick resistance and tolerance to heat is somewhat difficult, but we can potentially do that. And so rather than trying to measure all of those attributes in smallholder farmer herds, we would propose to form larger populations where all of the animals can be measured, where they can all have genomic information. Now, in Australia, we have done that in Northern Australia because we have cattle breed societies, and those cattle breed societies have come together with the input from our research and development partners, and they have funded the formation of these reference populations. In Africa, again, with government assistance, but with input from the smallholder farmers, we've been able to form populations that are genuinely representative of the sheep and goat and the cattle populations in Africa. There has been, in Africa, we've been fortunate that the Bill and Melinda Gates Foundation have been able to fund some of the work in cattle. So I'm not seeing that this is going to be funded entirely by the smallholder farmers, but it needs to be done in partnership with the Indonesians. We are already working with 3 Indonesian universities, and you know, I don't want to name names here, but those staff members from those universities have expertise. They have been trained in genomic selection, so they understand what we're needing, and you know, they understand the cattle breed populations. You know, so we are thinking we would work initially with Bali, Ongol, Madura. Clearly, there are lots and lots of crossbred animals in Indonesia that you know, the breed composition is just totally unknown. And so we would have to use genomic information to identify those cattle. But I think the way forward is through collaboration. So finding the people that are interested in becoming involved in this type of approach would be the first step. We already have contacts. I've shared an example with the potential collaborators where we have pooled our data from Brazil, from South Africa, and from Northern Australia, and delivered genomic predictions on tick resistance, which is probably one of the most difficult traits to identify in cattle. And so we've proven the approach works, and those collaborators, those reference populations are very interested in sharing or pooling data across other reference populations in tropical areas, because it's the tropical areas that are creating the difficulties here. So I don't have an immediate answer, but certainly I think we have interest and we have some keen collaborators who are willing to work with us. And one of the questions was from a farmer who has a small farm and is wondering how to get involved. So I think there's huge potential.
Okay, thank you, Professor Barrow. Now to Dr. Min. The question is from Nurfianto. How to this technology to be applied by farmers, and does this technology affect the livestock health? Do you get the question, Dr. Min?
Yes, sir. That's a good question. I partly answer at the chatting room. In tropic, can you hear me?
Yeah, it's clear.
Yes, in tropical area like Indonesia, there are lots of natural resources such as mimosa trees, acacia trees, cocoa, and various teas that contain lots of natural ingredient polyphenols such as tannin too. So that actually protect animal health such as bloated and acidosis, and you can modification of microbiome in the rumen. However, if you fed too much tannin, which is a negative impact, so local farmer can utilize natural resources such as tree leaves, mimosa trees, and acacia. Especially goat producers, they love to eat that kind of tannin-containing forages or forbs and tree leaves rather than white clovers and red clovers. So, which is pretty good opportunity to utilize natural resources nearby farm bases.
Okay. Probably if there are more questions, I will back to you if the time is we have still time. Now I want to go to Dr. Okalahan. This question is thank you for the informative presentation. Are there any possibility for the atypical Brucella, which mostly misidentified to the spread and found in animals? And are there any potential to emerge as MR Brucella? It's a question from Harimurti, actually.
Yeah. Okay. Can you hear me? Yeah, good. So, so firstly, to answer Susan's question, these strains, so far, they grow very, very rapidly. You can put them in CO2, you can put them in, if you want, they grow they just don't look like normal Brucella, so you won't identify them as this. It's only during mass spectroscopy that you'll find them. At the moment, there are very, very few strains of atypical Brucella, so we don't really know how many there are, but we just have the idea that, in fact, if we start looking for them, we will find them. So this is the real problem. You have to look for them to find them. And a lot of them are coming from exotic animals. You know, one came from a ray, a sea fish. We have ours mainly come from frogs, for example. Frogs are a huge source. So perhaps, for example, in Indonesia, in the jungles and the rainforests of Indonesia, you will find a wide array of these new strains. Whether they are a risk for humans, for the moment, we can't say. Whether they're a risk for the livestock, for the moment, we can't say. But there's just something that has to be kept in mind. In terms of antibiotic resistance, Brucella is not normally antibiotic resistant. The advantage of Brucella, as I said, it lives inside its animal host. It doesn't get in contact with the sources of antibiotic resistance. There's no chances for it to get transferred plasmids or phages, whatever, with antibiotic resistance genes in. So antibiotic resistance is not a problem with brucellosis at all. There are 1 or 2 cases which have been described mainly in Turkey or in South America or in the Middle East, where they are getting some very, very low levels of rifampicin resistance. We've sequenced a large number of these with the Sanger Institute, and we're trying to work out what the genetic modifications are. But this is generally related to antibiotic use or misuse in livestock, where animals are kind of fed antibiotics. So this is something, again, we heard earlier about use of monensin, for example, but all these types of things, it improves the animal yields perhaps, but it is a huge risk of inducing kind of antibiotic resistance in bacteria, which are kind of far more common and could cause serious diseases in humans.
Okay, thank you, David. Now I will go back to Dr. Min. There's one question for you, actually. Which one? I can read. Oh, maybe. Maybe you can choose from here which one is one you, since you only have about 5 minute times. Is that clear to you, Dr. Min? The question is there, or I will read it for you. Did you agree that the source of methane emission is coming from high fiber source in the diet of ruminants? So what do you think if we formula the ration with only concentrate 100%, say like that? This is from Dr. Diki Pamungkas from Research Institute for Beef Cattle.
Okay. We can thinking about that. However, it practically is not possible for feeding 100% concentrate, because a lot of physiological problem in cattle, and because firstly rumination problem because of no fiber, only 100% grain, and also bacteria, especially Streptococcus bovis, that kind of bacteria troublemakers, they fermented so fast in the rumen, they produce lots of gas, so make frosty blood. Also, suddenly, that kind of bacteria produce lots of acetic acid and other hydrogen. So they make a very low rumen pH, like acidosis. So animal got problem if we feed too much concentrate, even methane gas reduction. So optimum level of feedlot calorie in Texas normally 60% to 40%, depending on feedlot management. About 60% grain or 62% grain and 30% forages, fiber source, we generally mixing together and feed the cattle. So I think it is a good answer for him.
Okay, probably although we have the many questions here, but maybe you can answer in the spreadsheet and send it to us, because maybe the time is very limited. But ladies and gentlemen, thank you for all of the three invited speakers for very exciting presentation. And for the time being, the committee will give you the certificate. Please, for Professor Heather Barrow, you are welcome to receive the certificate. Please. Okay, thank you. And
Thank you.
for Dr. You are welcome. For Dr. Min, you are welcome to receive the certificate, please.
Thank you so much. Appreciate.
You are welcome. Dr. O'Callaghan, you are welcome to receive the certificate, please.
Thank you very much.
You are welcome. Ladies and gentlemen, this is the end of the presentation for the plenary session. Once again, let's give a big applause for all of us to having this presentation. To all of the invited speaker, please don't forget to send your full paper. We will put in the proceeding before the end of this month, I hope. Now back to Gracie. Thank you, Gracie.
Thank you very much for the speakers for informative and insightful presentation. And thank you for Professor Ismet for moderating this session. Ladies and gentlemen, we are going to take picture all of you for our documentation today. Please turn on your video and mute your audio. There are 5-page picture will be taken. Please be ready after third counting. For first page, 1, 2, 3. Next page, 1, 2, 3. Next page, 1, 2, 3. Next page, 1, 2, 3. The last page, 1, 2, 3. Once again, the next page, 1, 2, 3. It's done. Thank you. Ladies and gentlemen, it's already 11:30 a.m. and it's time for us for having lunch and prayer break. Coming up will be parallel session. For those who join the parallel session, please be noted that our session will be start at 13 o'clock Jakarta time. It will be great pleasure to have you to return on time.
Okay. Almost 400% come to the session, almost 400%.